My name is Jackie Imperial,
My name is Shreya Barma.
and this is our third and final primary research article. The title of this article is Signal Transducer and Activator of Transcription 2 Deficiency is a novel disorder of mitochondrial fission. The authors of this article are Shani et al., and the contributors for this research is the Mitochondrial Research Group. And the majority of this research was taking place at the UCL Institute of Child Health in London.
Before we examine this article further, let's go over a couple of definitions. The first being mitofusins, MFN1 and MFN2. Mitofusin is the process in which mitochondria of healthy cells undergo fusion. The course of action occurs in 3 steps. First, the mitochondria align themselves end to end. Next, the outer membranes of the 2 organelles fuse with one another. And last, the inner membranes finally fuse together, thus forming a larger mitochondrion. MFN1 and 2 are the proteins that allow this process to occur, especially within the outer membrane fusing. OPA1 is a protein that allows this process to occur as well, but is located in the inner membrane, allowing them both to fuse. The next definition is STAT2, which stands for Signal Transduction and Activation of Transcription. Febrile is an adjective that means related to fever, so in a medical sense, a febrile illness is one that involves a fever and is often used together with the word seizure. Now on to the abstract of the article. The first thing they talked about is how mitochondrial diseases are characterized. And mitochondrial diseases are clinically, biochemically, and genetically heterogeneous disorders characterized by the dysfunction of mitochondria, which are essentially just the cells that produce energy within the body. The mitochondrial networks are defined by a delicate balance of the fusion and fission events that allow essentially or link mitochondrial biogenesis to mitophagy, which is a quality control process for removing dysfunctional mitochondria within the mitochondrial life cycle. So this entire disease occurs when this balance is disturbed. And the next thing they talked about was the factors needed for mitochondrial fusion and fission. Mitochondrial fusion is mediated by several large membrane GTPases, the mitofusins, which I talked about earlier, MFN1 and MFN2 on the outer membrane, and OPA1 within the inner membrane. Factors that involved fission include the DRP1, which stands for dynamin-related protein 1, which is encoded by DNM1L, receptor proteins known as MID49 and MID51, and mitochondrial fission proteins known as FIS1, and mitochondrial fission factors, also known as MFF. Previously, mutations in the fission factors DRP1 and MFF have been implicated in causing human mitochondrial fission defects. And now they use a whole exome sequencing to identify mutations within the STAT2 protein, which encodes a component of the Janus kinase, also known as JAK. Now onto the materials and methods of this paper. They examined 3 patients. Patient 1 was the elder child of a non-consanguineous Albanian parents, who essentially non-consanguineous means that they were not related by blood. And essentially, this child was healthy until he was 12 weeks old, and then he developed a febrile illness 1 week after he took the MMR vaccine, and he was unresponsive to any antibiotics. His CSF cell count was completely normal, and he continued to develop a bunch of symptoms that led to a diagnosis of him having atypical Kawasaki disease. 1 month later, he was readmitted with opsoclonus myoclonus, and he got better for 2.5 years. And he was fine up until then, and then he was presented with more fever, diarrhea, hypoglycemia, impaired renal and liver function, metabolic acidosis, hyperammonemia, marked elevated CRP, and thrombocytopenia. And he then developed opsoclonus myoclonus and seizures with neuroimaging suggestive of meningoencephalitis. Patient 2 was the younger sister of patient 1, and unlike patient 1, she was healthy until she was 13 months old. She developed a febrile illness 1 week after the MMR vaccine, just like patient 1. She was anemic and lymphopenic, with deranged clotting. She made a slow recovery with no neurological or other deficits, and she remained well other than having some diarrheal illnesses, during which immunological testing revealed marked lymphopenia with low B and T cells. Patient 3 was unrelated to patient 1 and 2, and she had reported febrile illnesses, and her affected cousin developed a sterile encephalitic illness following the same vaccine, the MMR vaccine.
The conclusion ultimately reached through the study is that there are interactions between immunity and the mitochondria of mammalian cells. This notion is suggested by the role of STAT's role in mitochondrial function, specifically STAT2, which proves to be a novel regulator of mitochondrial fission throughout various experiments. Furthermore, it is found that the manipulation of the JAK-STAT pathway aids patients with mitochondrial diseases as a therapeutic strategy. It helps patients by making small adjustments to the balance of fission and fusion of the mitochondria. Now I will discuss Figure 1 of this article, analyzing muscle and cultured skin fibroblasts. Figure 1a depicts images from electron micrographs of longitudinal sections of muscle on the top and fibroblasts on the bottom. These images were taken from samples of the patients and controls observed throughout this study. In the study, light microscopy of the skeletal muscle was shown to have no evidence of fission defects. However, under electron microscopy, it is revealed that from both possessed abnormally long mitochondria, indicated by the red arrow, which measured between 8 to 10 micrometers in length. This, therefore, suggests a major defect in mitochondrial fission, which is the issue tackled by this article. The top of Figure 1b shows electropherograms showing Sanger sequence, a method of DNA sequencing. It confirms that the novel homozygous stop-gain mutation is located within the SH2 domain of STAT2 in whole exome sequences in patients 1 and 2 is heterozygous in both parents. The bottom of the figure indicates that the mutation prompts a stop-gain at the amino acid position 612 and the STAT protein.
Now on to Figure 4, which shows transcript and protein expression of fission and fusion proteins and the DRP1 localization. Figure 4a shows qPCR analysis of DRP1, MFN1, MFN2, and OPA1 and the 3 controls C1 to C3 and the 2 patients 1 and 3. The samples were normalized to C1, and ACTB encoding beta-actin was used as an endogenous control. qPCR works in a very similar way to traditional PCRs, the major difference being that with qPCR, the amount of the product is measured after each round of amplification, while traditional PCR, the amount of PCR product is measured only at the end of amplification. The concept of qPCR is quite simple. Amplification products are measured as they are being produced using a fluorescent label, and during amplification, a fluorescent dye binds either directly or indirectly via a labeled hybridizing probe to the accumulating DNA molecules, and a fluorescence values are recorded during each cycle of the amplification process. The fluorescence signal is directly proportional to the DNA concentration over a broad range, and the linear correlation between PCR product and fluorescence intensity is used to calculate the amount of template present at the beginning of the reaction. The point at which fluorescence is first detected as statistically significant above the baseline or background is known as the threshold cycle, or a CT value. The CT value is the most important parameter for qPCRs. This threshold must be established to quantify the amount of DNA within each sample. It is inversely correlated to the logarithm of the initial copy number, and the threshold should normally be set above the amplification baseline, but within the exponential increase phase. Most instruments automatically calculate the threshold value of fluorescence signal by determining the baseline average signal and setting a threshold tenfold higher than this average. In the image of 4A shown here, they show the qPCR of each of the 4 prominent proteins, DRP1 and MNF1 and MNF2 and OPA1. And in each graph, we see that the 3 controls, C1, C2, C3, and patients 1 and 3, known as P1 and P3. Figure 4b shows Western blot analyses of 14 milligram proteins extracted with 1.5% N-dodecyl beta maltoside from control C1 to 6 and patients 1 and 3 fibroblast cultures. The blots were probed with antibodies raised against DRP1, MFN1, and MFN2, OPA1, MTCO2, and TOM20 as indicated. An antibody raised against beta-actin served as a loading control. Now, Western blots usually work in a way that is very unique. Western blots is often used in research to separate and identify proteins, and in this technique, a mixture of proteins is separated based on molecular weight and thus by type through gel electrophoresis. These results are then transferred to a membrane producing a band for each protein. The membrane is then incubated with labels antibodies specific to the protein of interest. The unbound antibody is washed off, leaving only the bound antibody to the protein of interest. The bound antibodies are then detected by developing the film, and as antibodies only bind to the protein of interest, only one band should be visible. The thickness of each band corresponds to the amount of protein present, and thus doing a standard can indicate the amount of protein present.
The conclusion reached within the study is that STAT2 is a novel regulator of mitochondrial fission and DRP1 is the master regulator. Because of this, slight modulations in activity of DRP1 causes mitochondrial phenotype and ultimately results in a defective mitochondrial fission seen within the patients observed. In the 3 patients who had elongated mitochondria, they harbored decreased levels of DRP1 at serine 616 and increased levels of serine 637. The findings of this study is extremely significant because it offers a novel therapeutic strategy of mitochondrial diseases through the manipulation of the JAK-STAT pathway, adjusting the balance of mitochondrial fission and fusion. Most importantly, this study is significant because it is relevant for common neurodegenerative diseases such as Alzheimer's, Huntington's, and Parkinson's disease. These are our citations. Thank you for listening to our presentation.